Hi everybody, it's a great pleasure to be participating to this nanoscientific symposium on nano applications for today's changing world. My name is Nicolas Durand. I have a PhD from EPFL based in Switzerland, where I studied nanofluidics. I founded Abionic based on the technology I invented at the end of my PhD, together with my colleague Ivan Murky, an expert in biomedical optics. We have been elected Best Swiss MedTech Startup between 2012 and 2015, and today are considering ourselves at a scale-up. The company is ISO 13485 certified, and we are 50 collaborators, including 8 PhDs. Based on nanotechnology, we have developed the world's most rapid CE-marked in vitro diagnostic platform and have the possibility to offer up to 14 immunoassays simultaneously with lab precision from 1 drop of blood and provide all results in only 5 minutes on site. Our vision is first to use our nanotechnology to transform point-of-care diagnostics for healthcare system, and second, to save millions of lives in sepsis today and in other infectious disease tomorrow using specific new biomarkers. To be more precise, we own a new sepsis biomarker called pancreatic stone protein, PSP, which indicates clinicians when to start antibiotherapy in case of sepsis, avoiding the unnecessary prescription of antibiotic treatment with the risk of creating resistance, and saving the patient life when antibiotics are actually needed. It is intended to be used at the bedside in the intensive care units, the ICU, the emergency departments, and of course the hospital's wards. Let me start with explaining the magic behind the technology of Abionic. On the left part of this slide, you see the standard method of quantifying proteins in a blood sample. It is called ELISA, and the principle is to wait approximately 3 hours that the proteins diffuse in millimetric environment where capture molecules has to be bound at the bottom. At Abionic, we force the proteins to enter into nano-confined areas where the diffusion distances to capture molecules are much shorter. In quantiseconds, in 2 minutes, we can measure similar density of bound proteins in our biosensors than after 3 hours as the usual ELISA test. In other words, and thanks to nanofluidics, we shorten the distances that molecules have to travel during the incubation times based on the principles of bringing surfaces very close to each other. On this picture, you see one of our biosensors. These are manufactured in a clean room, and my perspective is that nanofluidic biosensors has to be manufactured with a very stable materials such as glass, silicone, and metal. Plastics are not stable enough to guarantee long-term reproducible performances assays. Our solution offers very reproducible assays that enable low percent CV test and high sensitivity. We have demonstrated that nanofluidics substantially improve medical diagnostics by reducing time to results and blood volume, limiting or removing sample processing, and most importantly, showing performances in line with reference laboratory methods. Abionic succeeded in bringing together nanoengineering, biochemical, and medical sciences, and in developing unparalleled nanofluidic technology-based platform to offer patients the fastest point-of-care test solution using principles of traditional capture ELISAs. You might have recognized in the previous picture that we have designed our biosensors with the shape of a bottle. The nano channel being the bottleneck, we force the molecule to enter and fill the reservoir using a capillary pump. The surface of the biosensor is chemically treated and contains immobilized capture molecules. The patient samples containing the analyte to be measured is diluted with a solution containing fluorescently labeled antibodies that specifically bind to the analyte of interest. Through capillary action, the solution enters the biosensors where the biomolecules diffuse, interact with the capture molecule, and form fluorescent molecular complexes if specific. Capture fluorescent complexes are optically measured with a miniaturized microscope in the reading unit. The amplitude of the signal quantifies the patient analyte's level. Abionic's solution is based on the cutting-edge patented nanofluidic technology, accelerating biomolecular interactions in the biosensors, and the technology is miniaturized compared to laboratory equipment, and hence can be integrated in a point-of-care diagnostic setting. 1 drop of blood is sufficient, assay time is reduced from hours to 5 minutes, and no washing step is required. On this slide, I would like to share with you the molecular interaction dynamics inside the nanochannel of our biosensor. As you can see now, in the top right, the animation showing the rapid increase of the signal inside the nanochannel once the molecules are traveling through the bottleneck and inside the reservoir. We have here the lapse of the signal intake for 2 minutes. And as you can see, after 2 minutes, we reach the peak and the saturation of the signal. We can observe a predominant capture of the signal at the entrance of the nanochannel, and here, a highly sensitive dose-dependent nanofluidic protein interaction. Our reading system is measuring the signal after 2 minutes and provide the user with a quantification immediately of similar quality than the one he will have received from an analysis laboratory after several hours or days. Up to 14 biosensors are disposed in a capsule, which is the name we gave to the single-use consumable depicted on the pictures on the left. The drop of blood is deposited onto the pad of the capsule with a proprietary pipette, and the capsule is inserted into the abioscope, our reading system depicted on the right. The abioscope reader has a user-friendly interface and is compatible with hospitals' patient data management system. The user pilots the abioscope with a big touchscreen, and it has been designed as a benchtop device for hospitals, intensive care units, emergent departments, ward, but also for primary care with physicians' practice and pharmacies. We also developed a smartphone and tablet compatible application to capture the patient data and guide the physician through the diagnosis. It is extremely useful in the field of allergies, where we propose capsules containing several specific immunoglobulin E, IgE, where the app is proposing cross-reactivities to make sure that the allergist is discovering all allergens. From one drop of blood in a few minutes, the patient is diagnosed and get out with a comprehensive report branded with the hospital's or the physician's practice visuals. In other words, thanks to nanotechnology, we have developed the only in vitro diagnostic point-of-care system that uses proprietary nanofluidic technology on one drop of capillary blood to analyze up to 14 parameters simultaneously in 5 minutes. Now let me introduce you to the most important indication where time is of essence, sepsis.
This is Tom. 3 days ago, after a simple infection got out of control, Tom's mother checked into a hospital. She had sepsis, a life-threatening condition arising when your body overreacts to an infection, causing injuries to tissues, organ failure, and may lead to death. Sepsis can strike anyone at any time. It requires immediate diagnosis and optimal clinical management. Every 3 seconds, someone dies of sepsis. Today, doctors face numerous difficulties to diagnose sepsis early enough. Now, help is on the way. The Abioscope platform enables early and effective detection of septic patients from one drop of blood in just 5 minutes, saving millions of lives, like Tom's mother. Abioscope, 5 minutes to save lives.
Sepsis is known as being the second cause of death worldwide, just after the cardiovascular disease. It kills more people than every year all cancers together. It represents 11 million lives a year. The number of deaths due to sepsis has dramatically increased in the last 2 decades, mainly because the fear of antibiotic resistance that leads physicians to wait much longer before prescribing an antibiotherapy. Every 3 seconds, someone dies from sepsis, and the mortality increases by 8% every single hour that the antibiotherapy is delayed. If diagnosed with sepsis and treated within the first hour, the patient has more than 80% survival chance. And we have identified that our technology could be a fantastic solution to diagnose sepsis very fast. So we have been looking for the best sepsis biomarkers, and one caught our attention. It is called the pancreatic stone protein, the PSP. This biomarker, generated by the pancreas and the stomach, is in permanence in our blood. However, the concentration increases when a severe bacterial infection occurs. As its performances observed in different clinical trials were slightly better than other biomarkers, we decided to acquire a license for its commercial use. In the current septic patient's journey, as depicted on the top part of this slide, the standard of care is detecting sepsis late, leading to many deaths or long hospital stays with long-term complications. What if we can offer the possibility to diagnose and identify sepsis much earlier? And this is exactly what we observed in several clinical trials. PSP is raising much earlier than any other sepsis biomarker, more than 24 hours earlier, up to 72 hours in very clean populations, such as severely burned patients. And this is huge if we keep in mind that the patients lose 8% survival rate every single hour the treatment is delayed. PSP acts as an early warning indicator of sepsis, which helps physicians prescribe antibiotics at the right time for the right reason. We have demonstrated the value of the PSP on our nanotechnology with a European clinical study in 14 sites in France, Italy, UK, and Switzerland. We have been able to demonstrate the fantastic value of identifying sepsis 24 hours earlier. For example, here, we see the case of a 46-year-old patient admitted at ICU for brain trauma. This observational study shows on the right part that the gold standard did not raise with sepsis, that the standard of care diagnosed sepsis at day 5, too late, and unfortunately the patient died from sepsis at day 8. The raise of the PSP from day 3 could have been an important indication for the practitioner to start the antibiotic therapy much earlier, allowing for better care for this patient and maybe allowing her to survive. And this is just the beginning. Sky is the limit, as we could potentially integrate any existing biomarker in our technology to provide clinicians with an immediate lab quality results from a drop of blood on site. This is only a question of time and resources to develop further applications in many fields, as you can see here. Our current ISO 13485 certified production line based in Switzerland has a capacity to produce in an automated way up to 1 million tests a year. We plan to duplicate this line in key countries in the future to facilitate the logistics and to support the commercial growth. The line has been designed to allow customization of each capsule, enabling personalized medicine and participating to the reduction of healthcare costs. In conclusion, our nanotechnology based on nanofluidics is the core technology enabling the world's most rapid diagnostic platform. Used in conjunction with the best biomarker for early detection of sepsis, the PSP, it could potentially participate to save up to 11 million lives a year. With its highly versatile nanofluidic platform, Abionic has limitless expansion possibilities. I thank you very much for your attention. Enjoy the conference.